Good afternoon, everyone. Thank you for joining me this afternoon. I'm Mattia from Lonza Monteggio, our site in Switzerland, and I'm in charge of micronization and containment equipment sales all around the world. And what you see here, Lonza Engine, is a new brand that we have launched this year in June, which groups together all the equipment that we provide as legacy Capsugel, which joined Lonza a couple of years ago, and legacy Micromacinazione, which also joined Lonza a couple of years ago. So in this equipment portfolio, we have equipment for bioavailability enhancement and equipment that helps customers speed up through the clinical phases for the development of a new drug. So more in particular, for what concerns bioavailability enhancement, we have liquid filling and sealing technologies, which seal capsules without the need of a bending, but just by spraying a liquid between the two capsules to seal them. And then we have micronization equipment. Containment equipment is part of this, and this is what is going to be the topic today, because we have provided a very interesting prototype a couple of years ago that I want to talk to you about. Then we have technologies that help customers speed up through the clinical phases, and this is our famous Excelodose, which is a machinery that can feed capsules at pilot scale, let's say lab and pilot scale, with as low as 100 microgram of powder into a single capsule, which means that I can feed only the API without the need to mix it before with an excipient, and I save up to 6 months on the development of a new drug. Anyway, so today we're going to talk about bioavailability enhancement. And so this is our equipment portfolio for what concerns milling and jet milling and high containment solutions. So we have spiral jet mills, we have opposite jet mills, we have mechanical mills, and we have containment solutions, which means glove boxes, rigid glove boxes for very, very high containment levels. So very, very high means that the latest glove box that we have delivered, for example, is lined with a containment level of 1 nanogram per cubic meter tested. And this is, I would say, one of the highest in the world. And we are focused on very high-level containment solutions, completely tailored. So just to give you an idea, 1 nanogram per cubic meter means that only a few milligrams of powder are exiting, let's say, the glove boxes during an 8-hour operation. Only a few milligrams in the equivalent of a few milligrams in a volume of a soccer stadium. So this is working. OK, so today's presentation is going to be, again, about this case study. This is an equipment that we have delivered a couple of years ago. It's a prototype that we delivered in Italy. And for a for a micronization process at very low temperature with very special conditions and with a very, very high containment level at the same time. So the request from the customer, the need from the customer was to have, again, a micronization and a milling process contained to work on new chemical entities for the next 10 to 20 years. So we don't know which is going to be the containment level requested by the new molecules in the next 10 years, so we need to be as much conservative as possible. And also, another problem that they have with their APIs is that they're extremely hygroscopic, and they have a lot of amorphous content that happens during micronization. And also a lot of electrostatic charges, also due to the micronization process itself on their product. So their product is not so stable. Therefore, what we provided is a glove box able to work with a containment level of up to 10 nanogram per cubic meters, which means it's one of the highest containment levels in the world. Predisposed to work with a jet mill, a pin mill and a hammer mill on this side, on the right side chamber, and a conditioning process right after micronization. And I will explain you later why this conditioning process is very important and why this is sometimes linked to electrostatic charges, and this is linked to the micronization process itself. So before we start, just a brief introduction about what is an OEL, because we're going to talk about it, a lot about this during the presentation. An OEL is the maximum amount of powder of API that an animal can absorb without having an effect on the animal itself. This means that if I'm working on a high-potent compound, suppose for cancer treatment, for example, then I need to make sure that the compound I'm working on is not affecting my health, the health of my operator. Therefore, there is a certain limit that I can breathe, that I can absorb during my 8-hour operation, and I need to make a machine and a process which is far below that limit. So, in order to reach this very, very high containment level, we had to close the whole process into this rigid isolator. And you can see here where we have the milling chamber, the milling and micronization chamber. Then we had a conditioning chamber here on the right. And to enter and exit, to put inside and put outside a product, which is the most sensible step, let's say, of the process, because it's the moment in which I open the unit and then I get out the product. So for that transfer, we used this airlock, this passbox between the main chamber and the outside, and a double continuous liner system, which is this pipe that you see here and the pipe that goes on the outside. And there is also a nebulization of water in between. So this means that whenever I want to introduce some product or some tools, for example, inside the main chamber, I have to go through a first continuous liner. So I do a first crimp and cut inside the airlock. Then I take my bag and I introduce it into the main chamber here. This chamber here or the other chamber, depending on what kind of process I'm running. And then I have a second crimp and cut so that this airlock remains clean at all time. On the contrary, let's say when I'm going out from the micronization chamber, consider that I have a product which is micronized, which means that it's very volatile. It's extremely easy that this product gets out and spills out from my bag, and so I need to take extra measures to protect my operator. So I'm going to pack my micronized product into a bag. I pull it out through the continuous liner. I crimp it, and then I wet all the surrounding surface of the bag. There is also the possibility to have a nebulization, let's say, of water to drop the particles so that these particles cannot escape from the glove box. And then I have my bag, which is going out again from this second continuous liner. So at the end, I have 3 barriers between me and the final product. And by this system, we've been able to reach the final containment level, and we have measured it, and I will show you later how we measured it. So now we moved into the micronization section of the isolator. The chamber, as was said before, was predisposed to work for a pin mill and a hammer mill, and 3 different sizes of jet mill. And the whole process was working up to -60 degrees Celsius, so a very low temperature, and the humidity was controlled inside this chamber. So just give you a brief introduction of what is jet milling. Jet milling is one of the most, I would say it's the most dusty process in pharma, and therefore the most dangerous when you work on high-potent compounds. So generally, I have a feeding system where I introduce my dry powder, around 500 microns, half a millimeter. The product is transported inside the jet mill itself, inside the micronization chamber, where it gets micronized. Then the air or the nitrogen is transporting my powder inside a second filter, which separates the process gas from the powder. The micronized powder, very fine particle, in a range between 1 to 3 microns for inhalers, for example, or 1 to 10 microns for tablets, is collected in the collection drums below here. So when I open, you have to imagine that when you open these drums, you have kind of a dust which is going out because this product is extremely fine. And if you look at inside, of the jet mill, you have the inlet of the raw material from the feeder. This product is transported into the micronization chamber through a push of gas. And then there is a second inlet of gas, which is accelerating these particles in a vortex. And within this vortex, the particles collide against each other. So you have the product which is micronizing itself. There is no crushing, there is no heating in this process. There is no mechanical contamination because you don't have friction between two disks. It's not like a pinmill, you know? And when the products are fine enough in this vortex that is created inside a milling chamber, they proceed toward the center because they follow the flow of gas and they exit through the top of the jet mill. And then they are collected below the cyclone filter or below the jet mill itself. So the two main parameters when I work on a jet mill are feed rate and pressure. So you need to think to the pressure as energy distributed in the particles inside the milling chamber. The higher the pressure, the higher the energy that I give to the particles. Therefore, collisions will be stronger, but my particle size distribution will be smaller. The problem is that the more energy I'm applying to the particles, the more chances I have to have, at the end, agglomerates because of electrostatic charges, or amorphous content, because if my product is not stable enough, it's sufficient to have a collision between two particles to change the crystal shape, the shape of the crystal of the particle. And this was the problem in this process. So other than these two main parameters that are normally used in micronization, we have also introduced additional parameters, such as the possibility to change the chamber material, which is the product contact surface. We can change the geometry of the chamber. We can change the amount of time that a product remains inside a chamber to influence the energy that I apply to each particle. Or we have also optimized the mill in order to avoid those common issues which are related with clogging or abrasion. Anyway, the micronization process in this case was running at minus 60 degrees Celsius. And I will tell you later the purpose for that. So anyway, the gas which is used to micronize was cooled down through the use of liquid nitrogen. So I have two serpentines inside this pipe here. And these two serpentines, in one serpentine, I have the gas which is entering inside my milling chamber. And in the second serpentine, I have the liquid nitrogen, which is cooling down the gas at minus 60 degrees. So the purpose for this is to make my powder more brittle, so that it's easier to mill it, to crush it, to micronize it, especially if I have water content inside it. And this means that to reach a very low particle size distribution, I can use less energy applied to the particle. Less energy, as we said before, means less electrostatic charges on the product and less amorphous content at the end. So this was one of the solutions that we adopted in this case to help the customer with this solution. So with their issue on their molecules. So we have one chamber which is working at minus 60 degrees Celsius, which is the micronization chamber. Then there is another chamber, which is the conditioning chamber, where I transfer the product after micronization. During micronization, it could happen that I have agglomerates. Sorry, after micronization, it could happen that I have agglomerates. These agglomerates could be soft, in case my product is absorbing water, for example, from the air, because it's micronized and it's easier to absorb water. And in those cases, it's sufficient to work with nitrogen. But when the agglomerates are hard agglomerates, like big stones at the end, which make my micronization useless, then that's mainly because of electrostatic charges. And these electrostatic charges are caused because of the collision between the particles and because of the friction of the particles inside the milling chamber. So there are different solutions for that. One of the solutions that we adopted, and that we adopted in this case, was the conditioning process right after micronization, which means that the customer is customer or us, when we do it in our GMP side, is taking the micronized product. They put it on trays, on vessels, stainless steel vessels, grounded, and they keep this product resting for 24 to 36 hours at a high humidity and high temperature. The high humidity is used in order to release the electrostatic charges from the product to the vessel. This way, my agglomerates are breaking apart, and then I can collect my micronized product without those big stones, let's say, inside the final product. So the humidity, so this conditioning chamber had a humidity control system, and the humidity control system was made by adding drops of water in the nitrogen. So we have made a recirculation system of the glove box, of the nitrogen inside the glove box, and we were adding drops of water to increase the humidity of this chamber. And to reduce the humidity on the other side, it was sufficient to condition and add additional nitrogen to inside this chamber. So again, this was used because thanks to the humid environment inside the glove box, the product is much more likely to discharge electrostatic charges to the trays. And all this is done inside the isolator. Then we have a heating of the same chamber. The heating is done just by electric resistance, so the concept is very simple. I have my nitrogen, which is going through these resistances, so it is heating up, and then it enters inside the chamber, inside the glove box. And we have, of course, an automatic temperature controlling system inside the glove box. And the purpose for this, the purpose for this is to make it easier to reach a very high humidity grade. So it's just related with the humidity, because when it's more hot, it's more likely to have 99% humidity at the end in the conditioning chamber. So after the conditioning is done, at the end of the process, at the end of the campaign, I have to wash the wall isolator, of course, to change to the next product. To do that, there were several spray nozzles located all around the unit and spray guns, so I have to wet all the surfaces. And then after I wet all the surface, I cannot anymore breathe in the product, so it's safe for the operator to open the front window and then wash manually the piece of equipment that they have dismantled into many small pieces. Otherwise, the other procedure, depending on the safety department, they keep the windows closed at all times. Also during the washing, they wash all the parts inside. They dry all the parts inside, and then they assemble again for the next product. And inside this unit, you can see at the bottom there is a small reservoir, a small tank. That tank was collecting the liquid used for washing this unit. So as said before, we've made this glove box with a 10 nanogram per cubic meter containment, and we have to demonstrate this. To demonstrate this, there is only one method to do that, and it's the SMEPAC test. SMEPAC test consists in putting a series of pumps all around the isolator on the weak points, for example, at the inlet, on the gloves, on the operator, and next to the outlet, next to the filters. And these pumps are equipped with a filter, and the pumps are simulating the human breath of a person during an 8-hour shift. So I simulate my whole process, including sampling, including washing, including the assembly again, including the whole micronization, actually. And I do this with lactose or with naproxen, for example. And these pumps are detecting at all time if there is any particle, any powder escaping from the glove box. So at the end, what you do is to collect all the filters in front of these pumps and calculate the amount of powder remaining on these filters. And by this, you can estimate which is the containment level of the glove box. So we have done the verification of this of the containment level of this isolator, and the minimum detectable amount on lactose was 2 nanogram per cubic meter, which is extremely low anyway. So by regulation, let's say, we have to make 3 shifts. So three runs, three trials. The first 2 with our the first one with our operator, and the second 2 with the operator of the customer, in order to prove that the training was actually successful and that in any condition we can reach this containment level. So we have a target of 10 nanograms per cubic meter, which means that we need to stay 30% below that value to be on the safe side, which means that the real target at the end is 7 nanogram per cubic meter. The minimum we could detect was 2 nanograms. During the first 2 runs, we couldn't find anything, which means that the contaminant level could have been 1.5 nanogram per cubic meter, or 0, or I mean 0 is really difficult, but 0.5 or whatever below 2. On the third run, there was a small mistake on a crimping, so on cutting the bag at the end of the process, and we saw, in fact, the containment level which was decreased. So there was kind of some particles going out, but still the value was below the target of 7 nanograms per cubic meter. So we successfully passed the test. So to summarize, this prototype was combining a micronization and a milling process at minus 60 degrees Celsius on this chamber. On the other chamber, there was a temperature of plus 45 degrees and a humidity of 99%, all with a measured containment level of 10 nanograms per cubic meter. So it's really a unique machine, a unique prototype, and this is the reason why I wanted to show you this prototype today.